Supported by an NSF fellowship to complete her PhD in biophysics in 1968. And that was actually involving work on phage, and I'm sure she's going to talk about that as well. She's taught and maintained a phage research lab at Evergreen College since 1972, which is just remarkable. And there was a recent conference on phage in Portugal, and half of the people that were attending had been to Betty's program in Evergreen.
At one time or another.
Yes, and she has put on 24 different Evergreen International Phage meetings, and is hoping to launch the last one at the end of mid-August of 2023. And this is the fellow I was telling about, Dr. Meyer, who you want to meet too. She's now a faculty emeritus at Evergreen, but she's still active in the field. And somewhere in this room is a wonderful styrofoam box full of phage that she has brought to our phage lab. So Dr. Fry, who's right beside me, make sure that that gets into the lab because these are the most prized possessions that Betty has been working on, and many of her lab folks have been working on for many years. So she's also the board chairman of Phage Biotics Research Foundation, which she started in 1997. So I'm just thrilled to have you here. Our IPATH is blessed to receive some of your phage, and some of these are going to make it into the arms and the bodies of people to save their lives. And so we'll keep you well apprised on that. So without further ado, Betty's going to be talking about the last 60 years of phage biology. And we have folks on Zoom as well. And we have muted your microphones until the end of the presentation. So great, take it away, Betty.
Well, thank you. And first of all, thanks for having me here. You've been, for a number of years, pulling things together in the phage field. And I remember the beginning of when you used phage to save your husband's life. And
there's the proof. The guy's right there, still picking.
And in the process of that, and in fact, I got reminded again about 9 months ago when a local book club called me and said that they had chosen the perfect predator to read and knew that I was somehow somewhat involved and would I be willing to join them. And I've been trying to join that book club for a while, and now they let me actually in.
And I actually forgot to just tell you that this is actually for you, dedicated to you. So you already have the hardcover, but now you have the paperback. So.
And thanks. You've done marvelous things down here in various ways that are one of the big centers that moves everything forward. And it's been amazing being at Evergreen. I'm rather isolated there to some degree, and it's interesting because 12 years ago I actually retired, but they allowed me to keep my office in my lab until this year, and they've just now closed it down. So it's a special thing to be able to talk some about these things in various ways.
Okay, well, we have standing room only, so we're going to try to, there's another chair here, if somebody can take the stuff off of that, and then, but we'll make room. We can slide down farther down here. I knew this would be well attended.
There's one more chair over there.
Okay, and there's one behind in the corner. There's a folding chair in the corner behind there.
It's up here.
Oh, Betty, maybe that's your stuff on that chair. Maybe we could.
Hi. Yes, that's my stuff.
Okay, so
let me grab it.
don't worry, Tom can move it. Sorry, but that makes more room. Yes, you can. Yeah. Okay. Sorry, folks. Sure. But it's great to see so many people here, which is great. Thanks, Betty. Sorry to interrupt you.
Hey, you're not interrupting. It's exciting to be here. And, you know, a number of times I've read what you were doing and so forth. And I remember that fall when you were just finishing things, somebody from your lab came and spent a couple of days with us thinking about it. But phage therapy is something that has long been something that was an exciting idea that in many ways its time had come. I did my bachelor's at the University of Rochester in Rochester, New York, and it's from there that they started setting up things to do phage therapy. And I wound up then, at the end of what I was doing, I took a few months off. Both of my kids were born while I was at the University of Rochester, and tended to go back and forth and be in my lab and so forth. One of the days when I came in, when Bernard was very small, he wasn't in his basket in my boss's office, and I walked in, and he was sitting in his infant seat on the counter, chatting with my thesis advisor while he was counting plaques. And that's the kind of support I got from John Weberg. And I'm telling this story because I want to say how important John was. He had just come there from being in Boston for a year as a postdoc in the lab of one of the people who a decade later got the Nobel Prize for work with phage. And all of that helped me and stimulated me during those years that I was working in that way. And being able then to have my baby come into my lab and be accepted and in various ways treated well during all those years was amazing. Do you want to grab any chairs from outside? I think there's some.
Yeah, sorry, we underestimated the number of people.
I need to tie down.
Okay. So John had learned from Salvador Luria to work with phage. He had basically been working there and arrived, well, my bachelor's I had done at the University of Washington in mathematics because that was what came easily to me. And I was actually top in my graduating class in mathematics, but it was boring. I had to do that. I had to finish it because I had to show that we as women can do mathematics and that it's an exciting thing to be doing. But it meant that that was what I focused on. But I got interested then in bacteriophage and learned about things that were happening at the University of Rochester and went to the radiation biology and biophysics department program at the University of Rochester, where my boss had just come from working with the phage people up in New England. And so I did my thesis on the transition from host metabolism to phage metabolism after T4 infection of E. coli. And with radioisotopes and so forth, did all sorts of studies related to phage then, and it was very exciting. That was at a time where there were not so many women doing things, and particularly not so many women with little kids. But my boss was amazing at letting me bring them to the lab when they were still tiny, and they were then used to being working with in the homes of other grad students while I was in the lab. We left there then to go to the University of Virginia when my husband had finished his physics degree. And I didn't try to get a job then, but I did do some phage work in one of the labs there for the next several years while my kids were really small. After the first few months of trying to be a full-time stay-at-home mom, I realized that wasn't me. And there was one of the labs particularly there that took me in under their wing and gave me a half-time fellowship to do that for the next several years. By the third year, I'd actually applied to NIH for my own grant, even though I didn't have any real position, and they had given it to me, and then I got a second one. And I went to talk to the chairman department about applying finally for a faculty position. He got absolutely irate that a woman with young children would have the audacity to even consider applying for a faculty position, and said if I did, not only was I guaranteed not to get the job, they had 2 job openings, but he would no longer sign for my grant. That was the best thing he could have ever done for me. Because actually that was what made my husband agree then to leave there. My parents had been sending us information all the time from Seattle about this new school called Evergreen State College, which was just opening up, and that year it had actually opened in Olympia. And we decided we'd apply there to start things off right away. And it contacted them, and it turned out that just a few weeks later, a couple of their people were going to be going down the East Coast interviewing people in terms of jobs for that upcoming year. So we went down and had our interviews sitting on a park bench in front of the White House.
Oh my goodness.
Well, it turned out that one of the things Evergreen was really looking for was to have every possible mix of varieties of people, and a married couple both teaching with kids. That all of them, they offered us a job on the spot. We never applied anywhere else.
Great.
And a lot of people since then have asked if it wasn't strange for us to stay at this little Hodug college in Olympia, Washington, but it's been an absolutely amazing place. And I'm 83 now, and I formally retired over a decade ago. But until now, they had still let me have my lab and my office. And finally, the current provost decided it's time. And this has been a really hard spring and summer with knowing my lab was closing. But the fact of coming down here, doing what's happening, and seemed like a really strong thing to have happen. As a sign of what's going on. And I'll always be involved with Phage in one way or another, and in some ways do traveling. And just last summer, my student here went with me when we were invited to go to a big international Phage meeting in Portugal, and they paid all my expenses. And in the middle, they called me up to the stage and gave me an award for a lifetime achievement in Phage. I cried. And after that, particularly, there were 700 people there. At least half of them wanted individual pictures with me. So I didn't see much of the rest of the meeting. Anyway, that's a bit of my story of that part of what was going on. Let's think then about now some of what we've been doing and going through. These are basically slides that I've been using for a number of years, and a few changes. But we did have, when I went to Evergreen, very soon thereafter, the National Science Foundation gave me a grant, and then either NIH or one of those supplied me with a grant for virtually all the years I was at Evergreen, even though most labs there didn't have grants, but they believed in what we were doing, and they helped see. And we had 24 biennial international Phage meetings there with people from all over the world. Now the last one only had 55 people because that's when everything was in lockdown, and we couldn't do it on campus. But the last two before that had almost 500 people. And we're hoping this August to restart that, and at least once more do that kind of Evergreen Phage meeting. Are there any of you that would be interested in coming to that? Significant number. Anyway, it's been my life in a lot of ways. I'd like to talk a little bit about some of the kinds of things my students have been involved in studying over the 50 years that I've been there at Evergreen, with the only basic research lab that was funded by always either INH or NSF in the school much of the time, and at most a couple of other labs had something. The T4 Genome Project was a major thing that we did working together with Bruce Alberts, where I'd done a year of sabbatical at UC San Francisco. And the genome was completed and published in 1994. We later on found one mistake. It was something that was done with the help of Bruce Alberts and the sabbatical I had spent down there, and with the help of other people from various parts of the world. And I should mention, by the way, that when we were at this meeting in Portugal this summer, seeing that over half the people had been at least once to Evergreen, I realized we really have to do more to keep it up. I'm not sure I can promise beyond that. And I'm mentioning that partly because I think you guys need to take over something like that. You're a place that's right in the middle of the whole phage field. The T4 Genome Project was exciting, and I had done the sabbatical with Bruce Alberts in the middle, and so we had done a lot of it then. It took several years actually working on it, but it was 1994 before we had the last gaps closed and published it. Work that happened later in other labs found one mistake in the whole sequence. So being able to do that, all with undergraduates, all in a college,
Before MinION ever existed for sequencing, right?
But, yeah.
Yeah, it was a lot of work.
Well, and a lot of it was by hand, but Bruce Alberts, who later on became head of the NIH, who I had done my sabbatical with at UC San Francisco, supplied us with money to help support it that year when I didn't have a grant, and B, helped with all of the things it had to do with finishing putting the sequence together. And there was a group in the Soviet Union who had also been doing work particularly on the tail fiber region and so forth. They invited me then to come over, and I wound up getting a grant to spend 4 months in the Soviet Union in 1990. And that was an amazing time to be there. Working with people at some of the various universities, which were places that, the one other place in the world that had really been doing some serious sequencing, and basically all of it agreeing with what we were doing, and then reaching out to have things happen more back and forth.
May I ask, had the T4 phage isolate that they were using, had it diverged from the one that you were using?
By 7 bases. We found 7 different places where a base was different, which was amazingly similar considering the way that that was something that had been done for a long time. So that genome project was finished in 1994, and then we became very interested also, starting in 1990 when I did the sabbatical with Bruce Alberts, in looking at the T4 metabolism and looking at all of the different enzymes. And we worked with people in a number of different universities who were interested in different ones, and many of the T4 enzymes then started being available to be used for doing molecular biology work. 1990 was, I think, about the first time where any of those things became widely available. Then in 2003, I started working with phages targeting E. coli 0157, which was one that had serious health consequences. We had a version of it that was not bad, and we isolated 3 different phages specifically for that, which were used then by a lot of other labs afterwards.
I hope they're in that box.
Are they?
Yay!
She's the one who's
Those
been
have been
putting
my
everything
favorites,
in.
yes,
Thank you.
they're there.
No, we decided there were several places that deserved a copy of everything we had pretty much, and you were one of them. You were one of 3 in the
David's
country.
very happy about that.
So were they ON 57 specific and wouldn't plate on K-12 or
Right,
LB or?
yeah, these were ones that were specific for that.
And could you get host range mutants that would plate now on K-12 or B or some other, or CR63 or?
And I'm not sure, I think there was at least one of the other strains that they would grow on, but there were a number of them they wouldn't, but they did very well. I don't remember all the details.
Yeah, it's
It
been
was
a
a few
while.
years ago,
Yeah,
but it was something that came out very useful. Another thing that almost nothing had been done with before was to study phage infection of stationary phase cells. In general, it was assuming that it had to be rapidly growing. And so one of the things that we worked hard then was to set it up to actually find versions of the phage that would grow on these cells. And we did quite a lot of work, and most of that got published. One of the other things we've also focused on through the years are phage co-infections. What happens when you have 2 very different phages, or in some cases 4 or 5 very different phages infecting, and what advantages and disadvantages and special properties you have. That was great fun. And that's been one of our main things we did for at least a decade after 1998. And infection of the stationary phase E. coli, sorting out what was happening when it was in a hibernation mode, when it was not dead, but when it didn't have things that would allow it to actually grow. Those were fun. And then we decided that we didn't need to restrict ourselves to E. coli. And we started working some with Pseudomonas, particularly dog ear infections. Catherine Carrillo was working with us, and people at the Eliava Institute in Tbilisi. In 1990, when I went to the Soviet Union, I realized that the place where phage was really being worked on was down in Tbilisi. And I took a train down from Moscow to Tbilisi and spent some time there that set up a partnership that still exists. We have many people from Tbilisi who had come and work in our lab. Right at that time, when I was first there, of course, was one of the most difficult times for them, and the Eliava Institute was virtually shut down. But we managed to make connections there with several people and to help get a couple of grants that let us supply them with a lot. And we've collaborated with the amazing people who've been doing phage therapy work ever since the 1920s. And if any of you have a chance to go there, even just for a short time and a visit, or a long time to work with people at the Eliava Institute, do it. It's a life-changing experience. I found that anyway. And one of my students that I was closest with, Naomi Hoyle, wound up then going down to work there for a little while. There were several people from my lab that did that, but she wound up falling in love with the grandson of my best collaborator there and lived there for the next 20-odd years. And now with her kids are old. Now she's back in Washington for now, but goes back when she can. But there's been that ongoing bond between the Eliava and Evergreen through all these years. Then working with Pseudomonas, and there we got started doing that because one of the people in my lab had a dog with problems with ear infections. We decided to try them there, and it worked very well. And we then began working with a local veterinarian who had been an Evergreen grad, and that was fun. In 2009, we got involved with the whole question of cystic fibrosis and working with a total of 200 different cystic fibrosis strains. And that was the final year that I was actually teaching classes there. And a group of my students wound working with them, and there were 60 students who worked at isolating dozens of phage as part of their class assignment for my final quarter of teaching. And the other thing that we have that we share with a lot of people are the 100 T4-like phages and the ECOR collection, the E. coli collection of reference over many years, and complex patterns and so forth. And you can see that the different phages, these are all related phages generally, not specifically, I mean, T7 is here, T4 is here, all of the different regular phages, but there's an enormous difference in the number of different bacteria that they infect and ways of using them. And we've been sending out some of the ones we have there when people want to particularly work with further analysis of some of those. This is the person who's been working with me to largely help get them to people who would make good use of them. So she's been working with me for the last 2 years, on top of a few other duties. Anyway, the phage sensitivities show little relationship to other aspects of the bacterial phylogeny, which is what seemed very strange. The bacterial strains that are hit by CED1, for example, which is isolated from sheep, are what are the ones that are indicated by the vertical arrows. These are the lab strains. Most things hit the lab strains, but there are all of these other strains that we've gotten from different people working in the wild with them, and that have very different properties. And for those how many of you have done any teaching or are doing any teaching at undergraduate that involves undergraduates? Many of you do that. It's particularly turned out to be a really exciting way. The last time I taught this regular open class, I had about 40 students, and most of those tests were done by the students in that group. That was a main thing that we did with them over the course of that year. And that's just one of the kinds of things that it's really possible to do very safely and very inexpensively with students, where it's something that makes them feel like they are really scientists, and not just feel like, but be, and think about what they want to go on and do, and to what degree they want to keep on with those kinds of things. And it's been great fun. So then we've also, in recent years, been doing more work at studying what exactly are in the standard phage preparations from the Eliava Institute, which since the 1930s has developed all of this work, and we've had a close collaboration there, and people from there stay with us. And for example, and they have some, in addition, they have some cocktails that are therapeutic cocktails, like encophage, like SES, and so forth. And one of the things that some of my students have done is go through and check all of the different kinds of bacteria that each of those strains will grow on. Again, that's a wonderful student project that can give original and easy to do data, get students really excited about what they're doing, and at the same time give valuable results. So we use core strains of staph and E. coli and strep and so forth, and tested all of these different phage and encophage and so forth, and see which ones of those it hit. The intestiphage hit an awful lot of different ones. Purulent bacteriophage, staph, E. coli, strep, and so forth. And then the other thing was isolating individual autophages, simply getting local sewage and then trying to isolate bacteria from it, which would be sensitive to particular phages that we had isolated. And we've done, some of the students have done a lot of that kind of work through the years, and then they have something that really to deal with. And we've also worked with a variety of individual phages. But this is the Eliava Institute intestiphage kind of box. And I really, how many of you have done any work or had any contact with the Phage Therapy Center in Tbilisi? Just a few. I'd really encourage as many as the rest of you as can to think about trying to do that. And I've had several students who've gone down and spent some time there. And it's a fascinating place. I got at one point a grant to spend a number of months down there, and I feel like I'm sort of part Georgian now. Our recent students were using the same approach to determine the host ranges of coliphages that they were isolating from the Eliava Institute cocktail. And so these are some of the kinds of things that they've been finding. And this was the ECOR collection that was from the University of Rochester, looking at core enzyme gel patterns, these various different things. And for example, ECOR4 came from the Denver Zoo leopard, 1156 from Sweden, from all over the world. So we have some of those still. These have been older ones. I'd encourage all of you to try to get in touch with people, particularly at times from underdeveloped countries, and work together with them and build bridges that help the whole world extend the kind of things that you guys have done so much work with here. This is my first visit, by the way, to UC San Diego, and I've long wanted to do that, and it's been really fun and exciting coming here. Whoops, go back. Can you? Yeah. So this was looking at some of the different intestiphages, coliphages, looking at some phages assembled from the metagenomics with their closest gene bank reference phages. So you can see the gene bank ones at the top and the phages that we had been finding and looking at. There's lots you can do with individual phages all against the same bacteria and learn a great deal, and in some cases find ways that they can be useful, because phages are wonderful therapeutic things. Next. We worked with Gary Skulnick, who was at Stanford Infectious Diseases, and in 2001 in Bangalore, he said, my mother in 1948 was dying of typhoid fever before they had antibiotics. My father read in J-Bacti about a Los Angeles scientist who had discovered a phage that killed Salmonella typhi. He called up this guy that was flown up to us in Seattle. My father injected my mother in the hospital with phage, and the next day she was perfectly well. That's real infectious disease experimentation, a mix of science and daring and desperation. And that's a motto that I particularly am emphasizing here. Something to think about, about what all of that means. We only later learned there was much data that laid a solid foundation for this life-saving decision, not just the mouse work he was aware of. And from this 1946 paper, one of the most spectacular accomplishments was the rapidity with which the patient returned to his normal mental outlook. 24 to 46 hours, patients who had been comatose and in the typhoid state amazed everyone by cheerful, grateful attitude, asked for food, vociferously. That from 1946. A lot of these things have sort of gotten lost for most people in the literature. As we consider what should be required today for introducing phage therapies that have saved lives for so many years in Eastern Europe and France, it's worth thinking about this challenge. And I think, one of the important things that here you're gradually doing, and is happening in other ways, is to really build the phage community and the phage, keep working like you have been with people all over the country and with NIH and getting permission and so forth. And I also encourage any of you who are teaching to very seriously think about using phage, getting some phage, isolating phage and so forth to work with your students. It's something that's incredibly inexpensive in terms of what you need to do it. It's safe, it's fun. There are so many possible phages that they can find that it's something that's really doing something original. So we've had a lot of students through the years who, in various ways, she's been my major right hand the last couple of years while she was still coming back and going to school a lot of years after she had originally left. So let me introduce Ria formally.
I'm Ria Kaylin.
So a while ago, when I was giving a talk for the Phage Futures Congress over in India, I was thinking about what the important things were to include and to think about. Demonstrate the importance of exploring phage-host interactions under planned treatment conditions, not just generally, but what you have to do to use them for treatment. And a lot of people there in India had been doing that. And demonstrate the benefits of corporate and foundation support for college student collaborations in exploring the complexities of stationary phage, anaerobic, et cetera, infections by the bottle phages with examples of such work. And any of you who are teaching, phage are so easy to work with, and they're so fun. It's really a whole new world, and I've been doing that for the last 60 years, and I've never run out of steam. So this is some of the kinds of things. Infection patterns depend on conditions. All phages were tested so far, in fact, both aerobically and anaerobically, and very little has been done with looking at phage infections anaerobically, but it's quite easy to do. And using the Hungate technique, which is a technique in closed jars. But the infection patterns are often quite different, as seen here. This is Pseudomonas phage, Pseudomonas Evergreen 2, which is like phage N4, which is more published in PA01. And you can see that depending on the conditions, the aerobic infection looks very, very different from the anaerobic infection. And yet, when you're going to be using things therapeutically, a lot of what they may be seeing is anaerobic. And it's not hard to just use a Hungate jar sealed and pump the air out of it and do infections anaerobically. But I can't think of any other labs who've done extensive anaerobic work, and particularly for ones like Pseudomonas phages that may be used in that sort of fashion. So will a bunch of you promise to do some of those with your students? I'd appreciate that. There's the phage competition kind of thing. These are T4-like CEV1 blocks the synthesis of T5-like CEV2. But if you wait until the next day, the level of CEV2 actually is higher than the CEV1. In other words, the immediate effect is the one does better, but the ones that survive blow away in a completely different fashion. So there's so many kinds of experiments like that that give really important and meaningful results with things that are perfectly safe, and thinking about the students then can think themselves too about, gee, what conditions might be interesting, might be useful. And for them to do those becomes very exciting. And it, like T4, like CEV1 blocks T5 like CEV2, but then they switch. The CEV1's lysis inhibition allows the CEV2 to get the uninfected cells. It's because of the lysis inhibition in the CEV1 in that case, that it gets outgrown. Next. Some of phages will self-assemble on the inner surface of a cell membrane, like this. Phage spectra of infectivity aren't much related to other properties of the bacteria. Each phage binds to specific lipopolysaccharides and or proteins on the surface. T4 sees an E. coli B-specific lipopolysaccharide, but sees C on K12 strains. So use a lot of different bacterial strains, and they can be perfectly safe strains. They don't have to be pathogenic, because usually those safe strains are basically the same as the pathogens. They just don't have that one pathogenic thing. So if you take a phage or take a bacteria that is similar to interesting pathogenic ones in everything but the pathogenicity island, you can do all sorts of experiments. Here are a couple of my students who were using this approach to determine the host ranges of polyphages isolated from intestiphage. And here these two had never done that sort of thing before, but these were some of the experiments that they were doing then and showing off as we had posters up at the local meeting. Look how happy they are with themselves. To be able to do something unique at the same time that it's something that is part of a class of things that are broadly supported and straightforward, is one of the major things that I found wonderful and important. More specific features of T4-related phages: they have a tight, complex nucleotide synthesizing factory linked directly to transcription, and it makes the right ratio of DA and DT to DC and DG for T4 infection. And they turn off translation of the host mRNA as well as host transcription due to a slight Child-Dalgarno spacing difference, I think. And superinfection exclusion then occurs at 5 minutes. They won't keep infecting after that. So there are a lot of these little questions about phage-host interactions related to a dozen different kinds of factors that are involved. And you can have a whole class and have them have different things that they're doing and sharing with each other, and they're doing something original and something useful and something safe. So here in stationary phase, the host DNA is still broken down to single nucleoside monophosphates in the first few hours after infection, as seen as in a DNA negative amber mutant. The nucleotides being released are still phosphorylated to DNTPs and incorporated into new DNA in the wild type phage infection. Now, there are all sorts of questions that you can think to ask, and that students can think to ask too, but with whatever combination you're taking. Then T4 has evolved to make the nucleotide synthesizing complex to synthesize DNA in a specifically efficient fashion. And this is the protein of the T4 thymidylate synthase. We've isolated some of those proteins and gotten somebody to do EM stuff to give us the structures. And you can see the complexities of it. And this is the sort of thing that's going on, where you have the tightly coupled nucleotide synthesizing proteins synthesizing the complex that then feeds into the replication fork. So there are a lot of physiological kinds of experiments that most people would never think of doing, that students can do easily. And then, get somebody to do the crystallography and look at it and see where the hydrophobic is. For example, these are both thiamidolite synthase, but looking at the difference between the host thiamidolite synthase and the T4 synthetase, there are quite significant differences in structure and the way it's put together. Next. This is one of the case studies that was done. Alfred Gertler from Toronto had a badly smashed ankle. The staff was still draining from both sides after 4 years, a full year on an IV antibiotic. Yet his staff was still sensitive to that antibiotic. And he went to Tbilisi, and there they perfused it with liquid phage for 24 hours. Strips of phage bioderm were inserted next to the bone. Within a few days, no more bacteria were seen in the exudate. There's, a lot of that sort of thing that goes on in Tbilisi. And think about it and think about doing those kinds of things here with students too. Next.
The next photo is a little more graphic, I think.
So 68-year-old diabetic with severe vascular insufficiency, post-steading with necrotic toe following distal embolization. And these were what was happening there, and it completely healed. And this was this work was done by a podiatrist who was my student back in the 1970s and is on our board of our Phage Biotics Research Foundation, and has now made a fair bit of use of phage, and he's doing more work this year and putting them back in much more, using different phages for these foot infection kinds of problems, and he's had a lot of amazing successes with it. Working through the pandemic, my recent lab graduates in Curtin, Explories, Maria Kalin, Chelsea Anderson, and Amanda Pratt have screened 46 unique phages that they've isolated, mainly assessing their differences by variation in host range and/or EOP. They're still finishing up their work with them, including genomic annotation using Patrick and Galaxy. This collection of phages are now being delivered to colleagues at Cornell, IPATH, and Phages Australia for further development and potential therapeutic and environmental applications. We'd be glad to share them. Kutter, yeah. And phaging on. Currently, there are over 40 global entities that are working with high hopes for swift advancements in biotechnology, biotherapeutic, and manufacturing. These advancements aid in safeguarding humanity by developing precision targeted medicines and treatments. And these are some of the different people who are doing things with it. And we encourage all of you to think carefully about that and think about doing basic undergraduate research kinds of things in ways that have true long-term meaning. Thank you.
Awesome. Well, we have about 15 minutes left. My counterpart at IPATH, who I didn't introduce at the beginning, is Dr. Chip Schooley. We actually have a couple of other IPATH faculty here, Dr. David Pride, who leads our lab, and Dr. Saima Aslam, who's treated the majority of our patients here.
That's you.
Yes, that's right. And then there's lots of students and faculty. This is actually Dr. Justin Meyer, who I told you about, who's doing his lab has been training phage. So there's going to be people that have lots of questions. Chip, did you want to moderate the Q&A for the next few minutes, or?
Sure. We'll also join Stephanie with our gratitude for your spending your afternoon with us and all the advice you've given us over the last couple of years and what you've done for the field over the last 6 weeks. Your work will live on here and other places. So thanks for everything you've done. So questions from people.
Oh, and there's some folks also on the Zoom. I actually see some faculty on the Zoom too.
Okay. So I'll try to watch the Zoom for hands.
Okay.
So anybody have any questions or things that Doug?
Yeah, so inspection of the stationary phase coli is really interesting.
Yes.
Do any of the phages that will infect, do they actually carry out a full lytic cycle during stationary phase, or do you have to?
There are some that will make bursts of half a dozen instead of 500. in stationary phage
And I'm
cells.
just thinking that if you had, so that's great having that lead. I'm
Yeah.
just thinking of, I'm guessing that some of the kind of bacteria that form vegetations on indwelling devices
Yeah,
in human bodies, the bacteria may be pretty stationary. And it
Yeah.
would be wonderful to kind of select for phages that would infect
that
those
will,
pathogenic
that will be
bacteria.
able to kill them,
And
yeah.
it would actually, you know, and probably by I could imagine cycling, you could get variant phages that would now grow much better.
Exactly, and that's one of the things I was kind of trying to emphasize. Don't think it's just simply the lab strain
Oh, no,
or
no, genetics is great.
whatever, yeah. But you can isolate things, in fact, by doing them, infecting that kind of thing in such a way that the progeny will be primarily the ones where they could grow better.
Mhm.
So you can do all sorts of fun things with them. Yes.
Question. I'm sort of curious over the years, our lab here at IPATH, we do a lot of phage hunting. And sometimes we're just really successful at it. Other times, not so much.
Mhm.
I'm curious if over the years you guys have developed certain tricks for, when you have an organism where it's just particularly difficult to hunt for phages that work against them.
Well, one thing that often helps is to try having different combinations of things that you're growing it in. In other words, bacterium that doesn't grow very well in the standard medium, there may be ways to get it to grow more efficiently. And when that's happening, then you're more likely to get better infection. Sometimes it makes a difference if you take 2 or 3 days before you look for them instead of 2 or 3 hours or 20 minutes. There's some that grow very slowly, but as they do, they may make progeny that can grow faster. So it's really fun. Do you have any further suggestions about that?
I do, but I'll talk to you privately maybe, because it's a long conversation.
Sounds good.
Okay. You won't share it?
I mean, it depends on what it is that you're, I guess, going after would be, so that's where the conversation with me would start with what it is you're looking for and how do you get tricky with it.
Okay, yeah, we'll have that conversation.
Yes.
Elizabeth?
Yes.
So I mean, nowadays, we have obviously millions of different phage that have been sequenced and millions of different bacteria. Does anyone use sort of a machine learning approach yet to try to figure out which phage might infect which E. coli based on the sequences? I mean, there must be signatures that would allow you to potentially predict what is going to work with what instead of doing manual testing.
I've never tried doing that. I found that it's so easy to take a bunch of plates and have different bacteria on them and try spotting and so forth, and then try to figure out later why something worked better than other things.
Well, there's just so much sequence now with the microbiome and phages within the microbiome.
Yeah.
So,
I think there are groups doing that.
Yeah, there is a
developing
self-remodel that was just developed and published recently. But therapeutically, what we have is a problem with somebody like contacted us today from China, right?
Yeah.
And, this is somebody who has a gunshot wound with a very multi-drug resistant Klebsiella. And, he wants to get on a plane to come here to get treated. Well, no, hold the phone. First, we got to see, are you eligible? Second, we need the isolate because we don't have this technique readily available yet. So getting that isolate sent somewhere where the phage hunts can be done. So sometimes we have, that isolate sent to 2 or 3 different labs that are all doing a phage hunt
Right.
simultaneously. And so it really means that we lose time, and time means we lose the patients. So there's a real need for it.
It may mean you lose the patients, but,
Yeah,
but it's still worth doing.
Oh, yeah, but this, I think
Yeah.
the field is headed to like what John Paul Pernay calls the 3D printing model, right? Where you have the sequence of the bacterial isolate and then you either find the phage to match or you design the phage to match.
Yeah, Bob Blaisdell works with him, who was my student,
Yeah,
and,
Bob's company, sorry to interrupt, but the Sally Bioscience, and they're working on that. They actually have a machine.
Which one?
The
the sale, oh, yeah.
That, it's new, they've just come out with it. We met with them in Portugal, it's really fantastic what they're doing, and they have the machine.
Is that
and
a
it's-
Portuguese company?
No, they're in Belgium.
Oh, in Belgium, okay.
Yeah. So, you can contact them and see
Yeah.
what could be collaboration.
and Bob Blaisdell may know more about what's,
Yeah, Bob knows a lot.
Bob
about
was
things
my
like that.
student,
Sequencing person.
and is there.
We have,
But I'd say Belgium is definitely the center of all of that.
Yeah.
Sounds
like there's another question maybe on the Zoom.
so we have a question from Joe Bondinelli, UCSF. Joe?
Hello, can you hear me?
Yes.
Sure can.
Hi, thanks so much, Betty, for your talk. I was really interested in the T4-like phage diversity against the E-core collection, and maybe it's related to the sort of machine learning questions, but with the goal of being able to print or predict things really quickly, I'm curious if anybody, you or otherwise, has taken that T4-like collection and made some sense of why, which the T4-like phages have the broadest host range versus the ones that have the weakest host range, if there's anything there either through comparative genomics or experiments or machine learning to figure out what the differences are and then test those, see to predict host range against that collection.
Yeah, I think there's a lot left to be done there. I mean, we're down to a very small lab that the school is shutting down on us. And we haven't been able to do some of those kinds of things recently that we'd like to. So the more other people can pick up the pieces of thinking about them, about thinking about what other bacteria might be encountered, think about using different conditions, using stationary phase cells to get things that will work in certain work better often with things therapeutically, because often you have severe infections that really are stationary phase. They're just sitting there barely growing but continuing to. Do you have any other suggestions there?
But can I just clarify, that was done through traditional plaque assays, I assume, that panel. So it seems like there, even within a single experimental condition, you have vast variability between this family. And that seems very tractable even with standard approaches, to look at the accessory gene collection of those phages or to look at other tail fibers and other things.
Well, and I'd encourage you to find a nice big class of students who want to experiment and have them have the whole class then choose different variations, small variations in ways certain testing is being done. And you come up with, first of all, exciting things for the students, and secondly, something that really makes a difference. That would be hard to do if you were having to pay a technician for every one of those small changes.
Thank you.
Do you have either, do you know, or do you have hypotheses about what are the factors in the phage and in the host that make a difference, that give the phages specific for certain individuals of the E4 collection? I mean, is it tail fiber receptor interactions, or is it something after attachment?
We've
Or it's a
not in
variety?
recent years, but a few years ago, we did hundreds of those kinds of things, and it didn't seem to be obvious. So, I mean, we may not have tried enough different things, but keep track of what's
I
happening.
mean, I guess I'm curious, worse, did you do tests, for example, of finding, are there some phages which don't have productive infections in a particular host, but
Yeah,
still will
We have done some of that,
absorb,
yeah,
and even kill the host,
yeah.
but not grow beyond that?
Yeah.
So they might be specific to your chaperones or
There-
something like that.
There are an infinite number of those kinds of things that can be there, and it's too many for the number of people who have labs like yours, or even like ours. It's something that is a really good thing to get as many different schools, high schools, colleges, involved in thinking about all the different kinds of things that they can test. And there was the last year that I was fully running my lab not too long ago, there were a cluster of them of kids from the local high school who did all sorts of different things and learned things that I never would have thought to have specifically asked. That, there are many variants and there are many kinds of ways that can go. And it's such a wonderful, are any of you involved in teaching? And are any of you involved in teaching across boundaries to junior colleges or even high schools or anything like that? And if you aren't, put together some teams of volunteers to do a project with you, get some of your grad students to teach a class for high school students and get them to try all those. There's so many possible variants that the more people that you have trying them and thinking about them, the more likely you are to come up with things that make a difference. On top of that, it educates much more broadly a whole group of young people who likely are not ever going to take the kind of courses that you're used to teaching or doing that kind of work, getting as many as possible. And one of the things we did was to work with some local high school teachers. Various things, there are all sorts of ways to do that. And a number of small things we've come across that really make a difference that we would have missed.
Great.
Well, I think we're at the hour, and we really do appreciate your coming. And as I said before, this gift is
Yes, the
really
gift of
priceless.
Phage, the gift of life.
Priceless. The more important gift has been the gift you've given to the field.
Absolutely, and to your legacy.
So thank you so much.
Well, and amongst Betty's many things, she was a fact checker for the Clifford Creditor.
When you were first doing it, yeah.
Great, well, thank you for coming, everybody, and I'll hand it over to you, take your Phage box, and Betty and Rhea are available for the tour, and thank you, everybody, appreciate it.
Thanks.